Welcome to our channel. A modern commercial grower can dial their indoor tent to exactly 25 degrees Celsius or 77 degrees Fahrenheit, feed the plants a perfectly calibrated nutrient schedule, and harvest a crop pushing 30% THC. Yet for decades, seasoned consumers have been asking the same questions. Where did the deeply psychedelic, multi-dimensional highs of the 1970s go? And what happened to that noxious, eye-watering roadkill skunk smell from the 90s? The testing numbers are higher than ever, but the subjective experience is flatter. We are watching a mass extinction event of the plant's actual chemical diversity happen in real time. To understand what we are losing, we have to look at the original source code. In botany, a landrace is a wild population of a plant that has grown in an isolated environment for thousands of years, shaped entirely by raw natural selection. The most critical cannabis landraces evolved in the regions of Central and South Asia. The extreme temperatures, intense ultraviolet light, and arid soil of these mountain valleys engineered highly specific chemical profiles that the plants used to defend themselves. Long before commercial agriculture existed, indigenous populations cultivated, traded, and maintained these pure, unadulterated genetic lines. These isolated valleys held a perfectly adapted, uncorrupted biological equilibrium of cannabinoids and terpenes, a chemical diversity that the modern market has nearly driven to extinction. Walk into almost any modern dispensary, and you will be told to choose between an indica for sleeping or a sativa for energy. Biologically speaking, these are meaningless marketing terms that have nothing to do with the actual genetics you are buying. The true historical taxonomy is much more specific. Landraces from South Asia, scientifically classified as variety indica, naturally produced a high THC to CBD ratio, always greater than 7, and contained rare minor cannabinoids like THCV. In contrast, the plants from Central Asia, classified as variety afghanica, possessed a THC to CBD ratio below 7, and they synthesized completely distinct compounds called sesquiterpene alcohols. In the 1970s and 80s, commercial breeders brought these distinct populations to the West. Driven by a demand for faster flowering times and higher THC yields, they mashed these distinct lineages together in a process called introgressive hybridization. By repeatedly crossing the highest THC plants with each other, humans accidentally bred out the precise, diverse chemistry that made the original landraces so medically and recreationally profound in the first place. We can see the exact forensic evidence of this genetic bottleneck by looking at the disappearance of the classic skunk aroma. Modern hype breeders spent years hunting for plants that smelled like sweet fruit, candy, and gas. By selecting for dessert flavors, they deleted the skunk traits completely by accident. That pungent stench didn't come from standard terpenes. It came from volatile sulfur compounds, or VSCs. As breeders chased sugar genetics, the VSCs were entirely eradicated from the mainstream gene pool. This chart maps a massive study of the U.S. cannabis market. Because of continuous crossbreeding, commercial cannabis now collapses into just 3 dominant terpene profiles. This bottleneck funnels modern strains strictly into myrcene-dominant pathways, crippling the plant's aromatic diversity. Please like and subscribe. It really helps our channel a lot. The top-shelf strains sitting in premium glass jars today might have thousands of flashy marketing names, but chemically, you are buying the exact same stripped-down profile over and over again. This brings us to the core biochemical mystery. Why can a vintage heirloom testing at only 10% THC deliver a far more complex high than a modern clone pushing 30%? This flowchart maps the plant's actual biosynthetic pathways. A healthy plant utilizes complex enzymes to build a diverse array of cannabinoids. Crucial enzymes like CBDA synthase create balanced ratios. To maximize THC, modern breeding actively erased these specific pathways. Maximizing a single compound isolates the brain. Preserving the full chemical array triggers the Entourage effect, allowing a diverse spectrum of minor cannabinoids and terpenes to interact holistically with the human endocannabinoid system. The solution to this monoculture requires walking away from the hype market. It means we stop popping candy gas polyhybrids into standard 15-liter, or roughly 4-gallon pots, expecting a different result. We must actively hunt for true heirloom and landrace genetics. Growing these seeds is the only way to resurrect the lost complexity of his species. To experience the actual power of cannabis, we have to become seed keepers. We must preserve Mother Nature's ancient work before commercialization erases it entirely. Thanks for watching. This content does not constitute medical advice, legal advice, or recommendations for the use, self-treatment, or consumption of any substance.